Hi, everyone, and welcome to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. If you're experiencing technical issues, please let us know about those using the chat box or by emailing me at krogers@aspb.org. If you are having trouble connecting or need to leave the webinar early, know that a recording of this webinar will be made available along with all of the associated materials within a few days. If you have questions during today's webinar, please let us know about those using the GoToWebinar chat box. Today's talks will each be 20 minutes, followed by 10 minutes for question. Madeline Seal, who is an early career fellow at Leverhulme, University of Oxford, who studies changes in water status, will be joining us today to moderate the webinar. Maddie is also a plant physiology assistant features editor. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make this series possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. This webinar is part of our new virtual research talk series that we created in response to the closure of most universities due to the COVID-19 pandemic. Please visit our webpage for more information on this series and sign up if you'd like to be considered as a speaker. You can find us on social media at plantae_org and at ASPB. For those of you listening to this as a recording, feel free to reach out to us on Twitter with follow-up questions using the hashtag PlantaePresents. Thanks again for joining us today. I'm now going to turn it over to Maddie to introduce our speakers and moderate today's session.
Hi, everyone. And I'm delighted to introduce this mechanobiology special with both Liz Haswell and Naomi Nakayama. So our first speaker will be Liz, and she is a professor and HHMI scholar at Washington University in St. Louis. She studied at the University of Washington and at UCSF before working at Caltech and then moving to Washington University. She's also a senior editor at the Plant Cell, and you may have heard her as one of the co-hosts for the TapRoot podcast. And she's going to tell us about mechanosensitive ion channels. So over to you, Liz.
All right, you can see my slide and you can see my pointer. Is that right?
Yep.
Okay,
we can see
great.
it.
All right, well, good morning, everybody. I'm very excited to be here. I'm delighted to have a reason to change out of my PJs before noon. I want to thank the organizers for the invitation to speak here, for Mary Williams and Katie for running this whole program, and for Maddie for doing the moderating. So I know that I'm just here to help you fill your time until Naomi gets on. So I'm just going to tell you a little project that we're doing in my lab at Washington University in St. Louis. It is my group's long-term goal, really our dream, is to understand how molecules, cells, and organisms sense and respond to mechanical forces. And oops, oh, how do I go to the next slide?
Typically,
maybe down here.
In the middle of the slide helps.
Well, maybe
try to enter that.
There we go. I think I can't use that pointer at the same time. Okay, so mechanobiology, mechanoperception is actually a really important question because mechanical forces are both essential and ubiquitous. In fact, right now, whether you know it or not, as you sit in your chair listening to me, you are sensing and responding to multiple forces. Gravity is pushing you down into your chair. You can feel the chair underneath you, under your tushy, and you can hear the vibration in the air as I'm talking to you. If you were to get up and stretch, internal forces would keep your blood pressure even by controlling osmotic pressure and would adjust to the stress placed on your leg bones. So mechanical forces are also important signals for plants, and this has been recognized for a very long time. Even Charles Darwin in the 1880s studied the response of corn seedlings to a piece of paper attached to the root. And what he saw was that the seedling would grow away from the paper, and he concluded that plants sense and respond to touch. Now, we've come a long way since these seminal studies, but there are still a lot of outstanding questions in the field. For example, we're still not entirely clear how force can be translated into biochemical signals, how these internal and external forces are coordinated, how force perception scales from molecules to cells to tissues and to organs are all big questions. In my group, we're interested in sort of molecular questions at the very initial stages of plant mechanobiology. And we're particularly interested in how membranes and membrane proteins work together to sense and respond to mechanical force. And in fact, the plasma membrane, well, all membranes are actually subject to all kinds of deforming forces during the life of a plant. So from the outside, forces could such as, stresses, abiotic stresses like water availability, biotic stresses like pathogenic invasion, which you can imagine might lead to defects in membrane extensibility or cell wall integrity, and even developmental signals like cell division could impact a plasma membrane from the outside. From the inside, changes in turgor or again in cell wall extensibility happen during pollen tube growth, during lateral root emergence, during steps of programmed cell death, during guard cell opening and closing, and probably many other aspects of plant growth that we don't even know about yet. So all of these events not only impact the physics of the plasma membrane, they then in turn affect the proteins that are embedded in that membrane. And we have spent the last decade or so studying a class of proteins that are uniquely capable of capturing these physical deformations of the membrane and turning those physical forces into biochemical responses. And these proteins are called mechanosensitive, sometimes are called stretch activated, or sometimes force gated ion channels. So ion channels are just protein pores in the membrane that are gated and allow ions to pass across that membrane. The gating stimulus could be a ligand like a neurotransmitter, it could be transmembrane voltage, or as we're interested in here, it can be a force that comes from lipids. So as illustrated in this really simple cartoon, if you increase tension in the plane of the membrane, that's lateral tension, this generates a biophysical environment that favors the open conformation of the channel and allows ions to pass across the bilayer. So mechanosensitive channels thus provide a very clever mechanism for transforming force, which is perceived as membrane tension, into a biochemical signal that the cell can understand, which is perceived as ion flux. So I should say that understanding how lipid forces drive mechanosensitive channel opening is really one of the fundamental questions in this area, and is a place where there's a great current interest and a lot of research effort. Then the same can be said for what happens after the channel opens. Take a situation where we have a very simple cell, which I've shown here as just a circle. We're just going to ignore the cell wall for now. If this cell finds itself in a hypotonic environment, that is, an environment where the concentration of osmolytes outside the cell is lower than the concentration of osmolytes inside the cell, water will enter the cell and it will swell up. This will increase lateral tension in the membrane and would potentially activate any mechanosensitive channels that were embedded in it. So then three things can happen. So ions will go down the electrochemical gradient across the membrane, but the direction they go depends on the electrochemical gradient of the ion. So for example, a mechanosensitive ion channel could open and serve to release osmolytes. So for example, chloride ions would go out, would probably leave the cell. It could also create an ionic signal. This would likely be a calcium ion entering the cell from the outside. And then a third possibility, one that we're very interested in, but I won't have time to talk about today, is the possibility that a mechanosensitive ion channel could change conformation and lead to a non-ionic signal, a non-conducting function that is still activated by tension, but is independent of ion flux. As you might imagine, this is a very useful activity to have, and mechanosensitive ion channels are found in all kingdoms of life and in almost every genome that's been inspected. I wanted to talk briefly about the history of mechanosensitive channels in plants, just to give you an idea of how far we've come. So it's often forgotten, usually by animal biologists, that single cell action potentials were first described in giant algal cells. And during the 1930s, the excitation of squid axon and algal membranes were studied side by Cole and Curtis. Later on in the 1980s, the advent of patch clamp electrophysiology made it possible to identify a huge number of mechanosensitive ion channel activities in plant membranes from different organelles, different plants, and different cell types. Subsequently, we've leveraged the awesome power of Arabidopsis molecular genetics in order to identify a number of genes and proteins that are likely to underlie the activities described earlier. Right now, there are about 5 families of mechanosensitive ion channels in plant cells: the MISC-S-like family, the MCA family, the TPK family, the OSCA family, and the piezo family. Some of them are found in both plants and animals, like the TPK, OSCA, and piezos. The MCA proteins are found only in plants, and then the MSL channels are found in both bacteria and plants. So now we have a lot of candidate proteins to be studied if we want to understand how mechanosensitive channels serve plants. And so the next stage, I think, in the history of mechanosensitive channels in plants is to really understand the function and the regulation of these channels. And we're beginning to make some progress there. So just a little overview of what we do in the Haswell lab before I get down to the nitty-gritty. We're interested in how mechanosensitive channels contribute to mechanical homeostasis in the cell. We're interested in how they participate in mechanotransduction or signaling pathways. We're interested in the evolution of these different mechanosensitive channel families, and we're very interested in understanding their structure and how their structure confers the ability to respond to mechanical force. So today I'm going to talk about signaling pathway, and I'm going to talk about this one channel, MSL10. It's one of the MISC-S-like channels I described on the previous slide. Before I go on, I just want to make one point, which is that while I am going to be talking about MSL10 as if it functions in isolation, that's probably a not very realistic view. And instead, it's probably functioning in the context of many other channels that are present in the plasma membrane, potentially in the ER, in the vacuole, in the plastid, and in the mitochondria. And all of these channels are probably impacting each other, and how they coordinate with each other and with other components of the cell, like the cell wall, the vacuole, and the cytoskeleton is going to be an interesting study for the future. That being said, today we're just going to be talking about MSL10 as if it were operating in isolation. Okay, so first thing I want you to know about MSL10 is that it is a bona fide mechanosensitive ion channel, and we showed this in 2 different ways. First, we used whole cell patch clamp electrophysiology to show that it's required for the predominant mechanosensitive anion channel activity in root cells. So you take a patch clamp pipette and attach it to a root protoplast. You use that patch pipette to inflate the root like a balloon, and then you measure current across the protoplast membrane. And what you can see is that in a wild type root cell, as you increase pressure, you see an increase in current. If you do this in a plant that lacks MSL10 and 4 of its closest friends in the Arabidopsis genome, you're never able to get any current to pass. So it's required for that activity, and then it can also confer that activity on a heterologous system. In this example, we use Xenopus oocytes that are expressing MSL10, and we pulled an excised patch, a tiny disc of membrane here in the tip of the patch pipette. In this case, we apply suction to the patch pipette to deform that patch of membrane that activates any channels that are present, and then you get a trace that looks like this, where over time, as you apply suction and then release suction, you see individual channels opening and a stepwise increase in current, and then after the suction is released, you see a stepwise decrease in current. So MSL10 is definitely a mechanosensitive ion channel. We've known that for quite some time, but it's been very challenging to determine exactly what the plant is using this mechanosensitive ion channel to do. What I'm going to tell you about today is a phenomenon that seemed like a pretty good candidate, which is to serve as a cell swelling sensor. So it's been previously shown that hypoosmotic shock can lead to a range of downstream effects. The accumulation of reactive oxygen species, a reduction in endocytosis, the induction of a large number of touch and mechanoinducible genes, the nuclear localization of BZIP transcription factors and other downstream gene expression events, the induction of cell death, a MAP kinase pathway, and then a calcium transient. However, all of these different events, which were shown by a large number of labs, as I wrote down at the bottom of the slide, but it was really unclear, and still is, how and if all of these events are related to each other. In addition, it was proposed that receptor-like kinases, or potentially mechanosensitive ion channels, maybe wall-associated kinases, might be doing the perceiving, but that was, of course, unclear. So we hypothesized that MSL10 might be involved in the process. Before I get into that, I'm just going to give you a quick overview to what we know about MSL10. As I mentioned, it's a membrane tension gated channel that's non-selective with a slight preference for anions. So it's probably likely that it's mediating chloride transport out of the cell. It's expressed throughout the plant and localizes to the plasma membrane. And luckily, we have a couple of genetic tools at our disposal. We have a loss of function allele, which is a tDNA insertion, that's MSL10-1. And then we also have a EMS-induced point mutation that's a gain of function allele, a single point mutation in the C-terminus called MSL10-3G. And we know that MSL10-3G and other overexpression lines lead to dwarfing and cell death, but it was unclear what exactly that meant about native function for MSL10. So a postdoc in the lab, Debarati Basu, set about to try to test whether or not MSL10 is involved in cell swelling. So she put together this assay where seedlings, 5-day-old seedlings, were incubated in mannitol and a little bit of isoxaben. So we're both providing osmolytes and softening the cell wall. And then they're transferred into a solution that provides a hypoosmotic shock. So there's no mannitol at all in the solution. And then we do a range of different assays to test what happened. And so what we expect happens is that upon hypoosmotic shock to transfer to the solution with no mannitol, the cells would swell. This would increase membrane tension, activate any mechanosensitive channel activities, and then we could then measure the responses. And this is similar to assays that have been previously described, and just to front load a couple of controls without walking through them, isoxaben is an inhibitor of cell wall biosynthesis, but we saw all the same effects that I'm going to tell you about if we used a different inhibitor called DCB. We also saw these same effects if we used PEG instead of mannitol, and if we used really high concentrations of mannitol, we could also see these same effects. So we really think we're looking at cell swelling and nothing that's specific to mannitol or to isoxaben. And for everything, we also saw no effect when we treated them with an isoosmotic treatment. So if we transferred them into the same solution. So the first thing that she tested was to look and see what happens in calcium signaling. So we used plant lines that are expressing aquarin, and we transferred them. We actually used a plate reader here. So we injected water in order to dilute the solution and provide the hypoosmotic shock, and then measured aquarin luminescence. And what you can see here, this is a time course of calcium, a cytoplasmic calcium as indicated by a quoron. Here in red is a peak of calcium that you see in the wild type line that then goes away after very, very quickly. The arrow here is when the injection occurred. What was exciting was that in an MSL10-1 null mutant, this peak is very low, and the peak is much higher in the gain of function line. Now, if we do the transfer into an isoosmotic solution, we only see a very small peak, indicating that this is probably just the mechanical disturbance of the injection process, really indicating that MSL10 is completely required for this calcium induction. So then the next thing we looked at was gene expression. The Munchausen group had shown that a number of genes are induced in response to hypoosmotic shock in the root, and we wanted to find out whether MSL10 was required for this and whether we saw this in our assay. And in fact, we did. So if you look over here at touch 4 result, isoosmotic treatment didn't cause any increase in gene expression, but a hypoosmotic treatment, you can see a large increase. In the green bar, you can see that the MSL10 mutant shows less of an increase in touch 4 levels, and that by 60 minutes, everything is back down to almost basal levels. WRKE8 is another mechanoinducible gene. Here we see a sort of different dynamics where we see an increase in 30 minutes and an even higher increase in levels by 60 minutes of hypoosmotic shock. Again, MSL10 is required for the full induction, and the gain of function line, MSL10-3 here in black, we see a higher expression. We also went on to show that we saw increased transcript levels very similar to what we saw for touch 4 for the touch 1, touch 2, and touch 3 genes, and that again, these levels were positively influenced by MSL10. I don't have time to show you, but we also documented an increase in reactive oxygen species in response to cell swelling, and this was also potentiated by MSL10. As I mentioned before, we had previously observed that gain of function lines in adult plants had increased cell death, but when we were very interested to know whether we saw the same thing in this assay, and further to see if what we were observing might be programmed cell death. And so we did 2 hallmark assays. Here is a tunnel assay. This looks at DNA damage. DNA fragmentation, and I hope you can see here in the gray panel that there is a large increase in DNA fragmentation in the wild type here in red, but the MSL10 mutant here in green doesn't show any increase. The complemented line recovers this level of DNA damage as indicated by the tunnel assay, and the MSL10-3 gain of function line shows even more than the wild type. We saw a similar effect when we looked at caspase activity. So we were able to document an increase in these proteases, which are indicative of programmed cell death in the wild type, a decrease, less of an increase, I guess I should say, in the MSL10-1 line, and an even higher increase in caspase activities in the gain of function line. We also assayed a number of other hallmarks of programmed cell death, including a number of gene expression assays and cytoplasmic acidification. So I'm just going to skip this and go right to the summary here. So to summarize, I've told you about an assay that we developed for cell swelling of seedlings that involves cell wall softening in combination with a hypoosmotic shock. This treatment leads to a cytoplasmic calcium transient within the first few seconds, accumulation of reactive oxygen species within the first 30 minutes, and then increased transcript levels of mechanoinducible genes within 30 minutes. We also showed that this induces programmed cell death within 3 hours, and that all of these responses require MSL10. So given its function as a mechanically gated ion channel, we are proposing that MSL10 is actually the primary sensor for cell swelling in Arabidopsis seedlings. But of course, there's lots of questions remaining. I didn't have a chance to talk about it, but this whole process is regulated by phosphorylation of the N-terminus of MSL10, and we have no idea how that works. We're very interested in how cell wall and membrane tension might be coordinated in this pathway. We don't know whether this signaling is related to ion flux through MSL10 or whether this is a non-conducting function. We don't know how MSL10 promotes calcium influx. As I mentioned, it's more likely to be mediating calcium chloride release. So how it might be interacting with a calcium channel is an interesting question. And then I just wanted to talk super briefly about the real world relevance of this pathway. So when would a plant living out in the real world experience cell swelling? And one possibility is flooding. Another possibility is cell wall damage or water soaking that happens in response to pathogens. Just wanted to give you one little bit of preliminary data suggesting that MSL10 actually does modulate resistance to Pseudomonas syringae infection. So on the left, you can see at 3 days post-infection that the MSL10 gain of function line here, again in black, is slightly resistant to infection. If you look on the right-hand panel at infection with Pseudomonas dB29, this is a kind of a crippled Pseudomonas that's core minus. Now you can even see that at 3 days post-infection that the MSL10 null mutant is significantly more susceptible to infection. And again, the gain of function MSL10 line is more resistant. So we don't know whether this is true of other pathogens. We don't know whether this is a direct or an indirect effect, but we're very interested in studying pathogenesis as a possible real world function for MSL10's role in sensing and responding to cell swelling. So with that, I would like to thank my lab shown here, and of course our funding sources. And then I just wanted to put in a very, very quick pitch for the TapRoot. This is a podcast that's sponsored by Plantae, and that is I co-host with Ivan Baxter. We usually have a guest, we talk about one of their papers, but then we really try to dig beneath the surface of the paper in order to try to talk about the way in which the paper came to be, talk about the stories behind the science. So I hope you'll check that out on your podcast app of choice. And then now I'm happy to take any questions.
Thank you very much, Liz. That was a fascinating talk. So I'm going to just look at some of the questions that have come through. So a question from Sue Reed. She wants to know if there are other mechanosensitive channel families not found in plants and what they might be.
Well, there are a lot of mechanosensitive ion channel families not found in plants. There are a number that are found, well, there's some that are sort of related to the OSCAs, at least structurally related to OSCA channels that are now we know are found in the inner ear that mediate the perception of sound. So that's one really cool family. There's another set of channels that Miriam Goodman at Stanford, who I'm sure you know, studies. Those are involved in light touch perception in C. elegans, and I'm not sure if they don't exist in plants or if they just haven't been found in plants yet. So, and there are also bacterial channels that we don't find in plants too. Sorry, go ahead.
Great. Okay, so next question from Maria Florencia Ecoli. So the question is, do the MSL10 loss of function and gain of function mutants have any phenotype in Arabidopsis roots, seedling roots?
In the young plants, they look completely wild type. In terms of development, we haven't seen any, and we have looked hard, defects in, say, lateral root emergence. We haven't seen any developmental problems. They seem completely fine. Once the plants get older, and especially if they're grown in soil, then they start to just be smaller and more dwarf.
Great. Okay. Next question from Oliver Arts. So says that the mechanosensitive sensor could react to touch as well as cell swelling, and were you able to design the experiments in a way to make sure that there were no touch or other forces that might trigger these responses?
Right. Well, unfortunately, there's no way to do an experiment where there's no touch involved. But we did do an experiment where, so typically they were done where there were, the incubations were with swirling. And so we did an experiment where there was no mixing, and seedlings were just, so we do them in these little trays, and you can just pick them up from one solution, pick them up in a tray and put them into the next solution. And so we did an experiment where there's no stirring at all, and we didn't see any difference. But it's very hard to control touching when, like I don't know how you change out solutions without touching them. So it's complicated. But we tried to look at least the effect of mixing, and there wasn't one. Yeah.
Okay, thank you. A question from Ryohei Thomas Nakano. So there are many other stimuli that trigger things like calcium and ROS and programmed cell death. And do you know how much of the MSL10 mediated induction of those responses are specific to mechanostressors?
I mean, so for example, if we add FLS2, which is known to trigger calcium signaling, MSL10 is not required for that response, but we haven't really gone through like every possible stimulus and looked at MSL10 requirement there. So that's sort of that's definitely a good future direction for us.
Okay, great. A question from Thomas de Pepe. Is there some type of dose response effect, meaning that more pressure would mean more response?
That's a good question. We haven't really done that. We haven't done that experiment. That's a really interesting question.
Okay, thanks. And a question from Alex Costa. If you know if there are about any MSL10 interacting partners, or if you have any clues to that.
We have a lot of MSL10 interacting partners. It's very sticky. What we haven't found is any that we can validate with genetics. So yeah, we're very interested in who it's binding to, especially this N-terminus that we think is doing a lot of signaling, but at the moment, we don't have any binding partners that we can back up with any genetics. So stay tuned.
Great. A question from Vincent Chichoya. Are there any developmental plant responses to the activity of MSL10 in terms of morphogenesis?
Right. So in the young seedlings, no, but once they get older, then yes. So the older seedlings, when MSL10 is overexpressed, they are dwarfed. They have dead patches on them. They're they look like lesion mimic mutants. The MSL10 null mutant looks completely normal all the time throughout all of development. We've never been able to see any developmental defects at all in the null mutant.
Okay. Question from Zach Savage. Would you expect the force caused by a penetrating haustoria to invoke a similar stress to osmosis-induced swelling?
Yeah, that would be great. We are trying to test that now, because that's definitely like the picture you have of how a haustorium would form, that it could compress the plasma membrane or the protoplast, but I don't have an answer yet. That's a great idea.
Great. One last question, I think, from Stuart Sullivan. Could you expand a little on the role of phosphorylation in MSL10 regulation?
Sure. So just to summarize what we know so far, the phosphorylation of the N-terminus prevents MSL10 signaling. So I was telling you how these adult plants, when you have a gain-of-function line, you overexpress MSL10 or you use the MSL10-3 allele. In adult plants, they are dwarf and they have these dead patches. If you have phosphomimic lesions at these sites in the N-terminus, now you completely prevent all those phenotypes. So for MSL10 to do whatever signaling it's doing in adult plants, it requires dephosphorylation. In the assay that we studied, I didn't have time to show you, but in this seedling cell swelling assay, the same thing is true. So if we use a version of MSL10 that cannot be dephosphorylated, we do not get any of these signaling events. We get no programmed cell death. So we think that it's usually maintained phosphorylated to keep it from signaling, and then when it's needed, maybe membrane, well, our hypothesis is that membrane, our model is that membrane tension activates the channel, and that somehow leads to dephosphorylation of the N-terminus and allows it to signal. Yeah.
Great, thank you so much, Liz. That was an amazing talk and great questions. I believe that the full list of questions will be sent through to Liz as well.
Great.
So I think you can ask more on Twitter. Katie will know
For sure.
what the correct tag is. So we'll move on now to Naomi. So I think Naomi's having some issues with her video, but we should be able to hear her voice. Is that right?
Yes. Can you hear me okay?
I can hear you great. So Naomi is a senior lecturer in the Department of Bioengineering at Imperial College, London. She got her PhD at Yale University and was a postdoc in Lyon in France and in Switzerland, in Bern. And she was a Chancellor's Fellow at the University of Edinburgh, a Royal Society University Research Fellow, and is now at Imperial. I have a slight disclaimer, which is that a lot of the work she's going to talk about today, I also contributed to when I was a postdoc in her lab, but hopefully I won't be too biased in picking questions at the end. So I think that's all from me, so take it away, Naomi.
Thank you very much, Maddie, and thank you very much for the organizers for this wonderful opportunity, and the entire platform is amazing initiative. Yes, sorry about not being able to use the webcam. It seems to be a sort of a recurring problem for some of the Mac users. So some of you out there who might know how to solve this problem, please get in touch with Katie for future reference. Yes. So yes, hi, I'm Naomi Nakayama. My group has the name Biological Form and Function Lab because we study form, especially the structural form of a plants, and especially sort of and its function, structural function. We have two favorite systems so far we've been working on. One is Arabidopsis shoots, and the other one is the dandelion diaspore. And basically common theme is slender, elongated structure. And that's something, we are quite interested in because it is a, nope, can I change the slide? Yes. Because it is a very, very commonly found structural design feature in the biological world. Here I just copy and pasted a lot of different images that biologist of the turn of the century, Ernst Haeckel, illustrated of all sorts of organisms from all the phylum. And it doesn't take that much of flipping through his collection to realize that life on Earth really like to make cylindrical, elongated, hair-like structures of all scales on its surface. So hair-like structures, that's how I'm calling them. They are very common in the living world, and I would like to know, we would like to know why that is the case, and then. So it might not be surprising that they are so common because they are such a versatile and multifunctional structures. because it's a way to increase the surface area quite efficiently without needing to increase the volume that much. This kind of a structure would help some emission and absorption of a chemical exchange in and out of living tissue. And also because of the large surface area, it's good at catching things like water droplets. Also, the particular shape, especially when they are tapered, they can be bent very sensitively. So it's a very good structure for mechanosensing, and it's needle-like and pointy, so it's good for defense as well. So those are isolated sort of functionalities isolated hair-like structure can have. But when you bundle them in a pattern on the surface, multiple of them, you can confer even more greater variety of functionalities like needle bed, yeah, to scare away pathogens. Lots of surface structures could reflect light in a particular way. The huge surface area, it can be very useful for adhesion, like in Velcro that we are all used to. Also, they contain a lot of spaces in between, so it's good for packing, compaction of the tissue as well. Depending on the surface structure and the spacing, you can keep the water droplets away from the surface or down on the surface. And also the flow of air or media in general, it doesn't get through those very hairy surface as easily. So you can use it for thermoregulation, like we use a fur for, and our hairs can do that job. Also, it can have a quite interesting ways of controlling flow around it as well inside. All those functionalities, you know, it can co-occur, yes, and also they can evolve from one function to another quite easily because a tiny bit of changes in spacing and things like that could change their functionalities. So that's another reason why hair-like structures are interesting. We wanted to study more about how those hair-like structures interact, hair-like structures function, and we picked dandelion diaspore as our pilot model because so dandelion diaspore is one of the one of the most representative example of plumed seeds, diaspore. It has a plume or pappus, which is basically a bundle of bristles, like hair-like structures, sort of, placed on top of a seed fruit, and it helps to increase the drag and helps to for the dispersal of the seed. Yeah, the dandelion seed is one of the probably most iconic flying seed that is far to travel. Like in some cases, you know, travel over ocean, like a sea, and pioneer land and a pioneer in the sort of island. I used to live in Edinburgh, and we used to say like that local dandelion enthusiasts would tell us that, yeah, they are pretty sure that Scandinavian dandelion arrive across the North Sea to Edinburgh once in a while. And there isn't a good scientific literature how far they can actually fly, because that's a really difficult things to track, especially if they can fly, travel such a long distance. But the indication of it is, yeah, it's that we all sort of have this impression they are fantastic flyers. But this is, this parachute-like pappus, it's made of completely dead tissues, and we dogmatically think they act like a parachute because they kind of look like parachute, but in a closer inspections, no, they can't act like a parachute, not in a traditional sense, because 90% of its parachute-like surface is actually empty. So yeah, and so we started our study to understand how they get to fly so well by first capturing their structure really well in great accuracy. We did a micro CT scan and captured the 3D shape of it as accurately as possible. And yeah, we found a number of filaments, the hair-like structures tended to be about 100 plus minus 9. It was very consistent. Our samples tend to have about 1.5 centimeter in diameter, and those bristles, the hair-like structures, are spread a little bit in 3D, but mostly sort of spread horizontally. And that kind of a thing that we extracted the quantitative information out of the 3D scan. And we were trying to calculate how air would move using computational model, etc., etc. But proof of the pudding in understanding how media like air moves around the structure like papas is actually to visualize it, especially when you don't know how they would move from the previous studies. So we meaning Kahar Queens and yeah, rest of the team. So Kahar was a postdoc who led this part of the study. We made a bespoke tiny vertical wind tunnel, and it's a dandelion size. Put the dandelion. So this is how that diaspore looks like if you let them float above it. It actually moves quite sort of slowly, therapeutic. But if we fix them on top and then blow the same speed of air straight in the air as how the free-flowing the diaspore would experience at its terminal velocity, then you will see flow movement like this. This is a long exposure picture. In the close-up, they look like this. So I would like to draw your attention. There are a little bit of airflow going through the papas, but not as much as one would expect. But there are flow going around the papas and recirculating. This is a 2D shot, almost medium cut. So in reality, please imagine it's a ring of almost like a halo-like ring of this air movement, recirculating air, sitting right on top of or downstream of the papas, and they stay at the constant distance. So yeah, there are some airs going around the whole structure, indicated to us that this this parachute with an amazing amount of empty space somehow sort of preventing airflow to pass freely. But before going into that further, I would like to mention that this so that vortex ring, as fluid mechanicians would call it, this whirlwind, ring-shaped whirlwind, it creates a sort of air bubble of a low pressure zone. So that just sits right on top of it. Vortex ring is not a new sort of new thing, but like when you puff the cigarette, smoke, you can make, some people can make those like smoke ring. That's one kind of it. And also when you have a disk, solid disk, often the vortex ring would form right on top of it, but it's attached to that disk. But this kind of detached vortex ring that stays at the constant distance and then don't dissociate away, this was a first time to be observed. And so we happened to, by trying to understand how dandelion fly, we happened to find a new type of flow behavior And what was cool was this was so that low pressure zone would stay on top of dandelion, and that any object would like to move toward area of low pressure. So this could help the separated vortex ring, as we call it, SBR. SBR could help the dandelion stay aloft. Just with that. For almost 50 years or so now, that fluid mechanics researchers thought they know everything about all the possible mechanisms that could achieve flight or help flight. But this is a new type of flight mechanism, so that was also quite cool to find out. So some airs are not going around, going through the papas, so the papas is acting almost like not as porous as it looks like, or it appears to be, and that is because the porous media like papas, they have some they have this sort of phenomena called the wall effect. So when the air is, so the medium is really, so when the media is very, when the object is moving really slow and small, actually, the air is very, very viscous. And so you can imagine something like taking the kitchen strainer and then trying to push it down a honey. And yeah, it's very hard to actually have the honey pass through the strainer hole. Yes, so it's kind of like that. Individual drugs acting on individual filament structures would interact with each other and sort of reduce the porosity. So yes, we studied how the different porosity indicated by Darcy number would affect the drug, and actually when porosity is quite low, yeah, even if media the structure is porous, it can act like a solid disk. And then, yeah, when it's too porous, then everything passes through. And dandelion papas sits right above here, not quite when it becomes completely flow through. And so it can act like a little bit like a solid surface, even though it's really highly porous. And we try to understand or isolate the key design features that allows, it's enabling the formation of this separated vortex ring. Because dandelion papas is a quite complicated structure, if you will. Does it need everything, every features of this complex structure, or yeah, are there key determinants? So we had this hunch that porosity is important, and dandelion papas, it kind of look like a 2D disc. Yeah, if we want to really simplify, why don't we start from there, the simplest 2D disc with a different porosity? Let's see if the porosity, if its porosity is exactly the same, maybe that's enough to create this both separated vortex ring, and that we did. So this is a real papas, and this is the artificial papas that our collaborator Enrico Mastropaolo developed the method for to make it. It's made from a silicone wafer cut into really fine structure, mimicking dandelion papas, and we made a porosity gradient with it too. And so when we have this artificial papas with the same porosity, sure enough, we get the vortex, separated vortex ring, which is very similar in its 3D structure, the z-axis and also the radial sort of size of the recurring recirculating air was quite similar. And another way to characterize this the SVR behavior is that so SVR is very stable. It's symmetric, axisymmetric too, and stable. But when you increase the airflow and so on, it starts to break. It starts to become asymmetric. And then when that happens, actually, that flight of the diaspore will be quite unstable. So it's not good for flight anymore. And so, yes, we can actually identify for different disks of different porosity when that transition from a steady SVR to unsteady asymmetric SVR form. And that's what we identified for the gradient of a porosity, and so solid disk. And for porosity up to 4 or so, it's almost acting like a solid disk. And then from there, gradually, gradually, and then suddenly, that unsteady transition to unsteady, that particular, the point, the transition point shifts. And so the y-axis is called Reynolds number. A Reynolds number kind of captures, it's a dimensionless number, a very useful number in fluid dynamics to encapture the relationship between velocity of the media, the velocity of the speed of the object movement, and also a viscosity of the media it's moving in, and also the size of the object, because those things relate to each other and determine how that flow would behave around a structure. So the Reynolds number, the point in which that unsteadiness occurs. For the dandelion papas, we identified that point. So that's the green point, that real dandelion papas would transition to unsteady at that Reynolds number around 400. And then with, I'm sorry, it's the blue dots. So we looked at the multiple samples, and all of them, no, I'm sorry, I can't see the bits of the slide with the sort of webinar control panel, so I apologize. But it's the green dot that is indicating the transitions into unsteady. And then the blue dots are the different samples of a dandelion we looked at that at its terminal velocity, and it's all within the steady zone. So when dandelion diaspore are flying as it normally would, they would have a steady separated vortex string. I do want to point out though, those natural conditions, they are flying at the terminal velocity. They are within the steady flight zone, but it's not so far away from the unsteady so the transition to the unsteady state. And that because the dandelion papas does not stay open all the time. It actually closes when there's a lot of moisture around. So this is the work led by Maddie, our moderator today. So here is the series of dandelion papas movement when you put the 1 microliter of water droplet right in the middle, and within 30 minutes or so, it just slowly closes. And when they dry out this little bit of water, they open again. It's completely reversible. And in nature, often in a very wet cities like Edinburgh, it's almost like every morning when it's misty, the dandelion, the inflorescence would look like this. They are closed with lots of water droplets on top. And then, yeah, and then when we put them into the little moisture chamber we created, yes, within 30 minutes to 60 minutes, it captures the water droplets and then close, and it opens again when they are dry. Does this movement, the morphing of the papas, have a functional significance? Well, so we place the closed papas, closed diaspore into our window tunnel and observed. And sure enough, the SBR was much smaller and it looked more asymmetric. It's probably not a stable, it does not look like a stable SVR at all, and definitely the zone of low pressure is much smaller. And when Marie measured the falling velocity, yes, depending on the papas angle, the speed of the fall was much faster when it's wet. And remember that those transition from the steady state to unsteady state, the diagram, if we plug in all those changes of size of the papas and also the speed that they move, etc., plug into the that value, we see that the diaspore, the papas, they would be falling. Well, when they are falling, they would not have a steady SVR anymore. Most of them will be in unsteady phase. And then we try to see how that would relate to how well the diaspore would be able to disperse. And that's a very hard experiment to do in nature. And so Marie did computational modeling using meteorological data available from many different cities. They're available from 30 years into the past. She picked 5 different cities of very different weather conditions within UK, and learned the simulations. Okay, terminal velocity is this when it's closed. Terminal velocity is that when they are open, and all the sort of the point data about the humidity and the wind speed, and etc. And she simulated how far that diaspora would travel. So first of all, I would like you to concentrate on the 2 colors only. That one is that when the papas would morph, like how they actually do. That's the green graph. And the orange graph is when they don't do that at all. So that's a hypothetical situation, but they don't morph, they are open all the time. So we were a little bit scratching our head. We thought this mechanism would help the dandelion to choose the best timing for flying. And because when it's moist, usually the wind speed is lower, and that's not a good time you want to fly. If you want to go really far away, you want to pick the dry moment when the wind is really strong, and so they are choosing for best timing to fly. That was our first hypothesis, but it didn't really look that that much better to be able to move to travel far. And in the dispersal situation, long distance dispersal, it's often not the bulk of the seeds that travel far away. Actually, very few examples and travel far, and that they tend to make the difference, because even one pioneering plant landing to a new habitat could create new populations and so on. So we looked at the top 1%. Maybe it's only about top 1%. It doesn't look that different either. we are scratching our head. And we had to think about long distance dispersal a little bit, leading up on where dandelions are found, and also leading up on what it means to disperse far away. Yeah, we had a lot of sort of exploration of ecological meaning over this behaviors we are sort of observing and simulations are telling, but we decided to separate. Yeah, we separate the state of sort of when the dandelion diaspore would when they are closed, how far do they fly? Versus when they are open, how far do they fly in the corresponding fraction of the meteorological data they have. Let's separate and see how that looks like. And then there, the dry ones would go far, but most of the wet ones would land quite close to the parents. And actually, we found out most of them don't disperse away, even don't detach away from parent inflorescence when they are wet. So yeah, they tend to stay closer. That's true for the top 1% too, and maybe for the open ones, they do top 1%. They do fly a little bit far away. So and in search of the functional biological significance of this phenomena, that we led a lot into how plants as well as animals actually decide to how they make a decision to migrate their niche habitats, because it sounds like actually such a risky decision they can make. It's a romantic notion that we feel like plants would want to fly as far as possible to find farthest place to establish itself, but that sounds like such a bad idea, isn't it? You don't know what kind of land you are landing into, and the farther you go away from the niche, the habitat that your parents were actually successful enough to reproduce. I mean, that actually is probably a good bed for a good habitat to grow yourself as well. And so when you look at that kind of decision making in animal field, that there are a concept called informed dispersal, that actually animals, they don't really like to change their habitat unless it's really necessary. The habitat is poor in providing food or whatever it is. Only then they tend to move their habitats. And but this concept is not really commonly sort of known in plant field. But being a plant biologist, we all know how acutely they are sensing the environment and making a decision in their development and physiology to survive through that environmental stress, but also increase the chance of their survival and reproduction. So yeah, sure enough, they also, of course, sense the environment and sense the environment and use the information to decide the best timing for their dispersal as well. There are a couple of really nice examples, like dandelion.
No, I'm sorry,
Okay.
Naomi, I'm going to interrupt slightly because you're going a bit over time. So if you can wrap up, it would be great.
Yeah, just wrap up. Sorry, I apologize. So yes, so relative dandelion actually don't even make papas when that they grow in a moist environment. And yeah, dandelion seems to be using this morphing to stay in a moist environment if they're already there and travel if they are in a quite dry environment. And yeah, I'm terribly sorry. I really mistimed. And but yes, so we so my group, yes, just to wrap up. So yeah, my group actually is a quite interdisciplinary group. We use any kind of methods, theories, and yeah, assays necessary to understand why plant structures, what kind of functionalities plant structures have, and especially quite thematic design features of plant structures. And we use biology, physics, and engineering to untangle those those phenomena. And then was a sort of a pilot example of that. And so a very important slide I would like to point out. So this is Kahar, who led the flight mechanism part of the study. And this is Marie. You have seen her today. Yeah, she led the morphing part of the study. And yeah, we have quite a few intern students, and then that short-term sort of helpers who helped out with specific assays and so forth in the team, that highlighted in orange here from the past members of my group, and also collaborators, yeah, in Edinburgh, the three musketeers of groups that we work together, and Alexander and his supervisors based in Glasgow, and Anna Maria and Simone based in Aretsky's group in Lyon. We actually characterize the morphing, biomechanics of the morphing of the papas. So yeah, I think I should stop as soon as possible. I'm terribly sorry for going over time. How much over time was I?
Oh, no worries. I'm not sure how much, but that's fine. Let's have a few
Thank you
questions.
for your...
Okay, so we got a question from Noparat Anantaprayun, who asks what kind of techniques that you use to take the photos of the SVR.
So we shine seed of light with laser, and then we use the normal camera to actually, or the high-speed camera to capture the movement of air. Air had little oil droplets that is used as a flow tracer. So they would just float about in the flow of air, and then they would be, shining up in the laser, and then that's how we, follow the flow and then took the picture of the flow movement. Yeah.
Okay, thanks. And a question from Jeanette Schutz. How much of the airflow behavior has to do with the small size of the papas? If you made a large artificial papas with properly scaled weight and dimensions, would it still fly?
No, it's not. Actually, the Reynolds regime that I talked a lot about in the particular transition, steady transition. The Reynolds number, it's the that, or it only works for the combination, the ratio of those size of the object and then the speed of the object, how it's moving, and also the viscosity. So air, kinetic viscosity stays the same. So, a lot of people ask us, like, can we make a dandelion-inspired parachute for human? And we're like, no, you don't want to do that. Because, that when it becomes larger, the Reynolds number changes. And then also when you have a heavier things pulling down so you're traveling faster as well. All those things make moves your a bigger parachute into the unsteady domain of the diagram. So actually, from our study, what we found was that probably dandelion papas is the largest one possible, almost on the edge, to be able to fly with this mechanism. So yeah, not unfortunately not dandelion-inspired parachute for us.
Okay, A couple of questions from Suri and Oliver Arts asking a similar question. What is the mechanism behind the water-mediated closure of the hairs?
Yes, so that's a bit I couldn't share with you today, but we are definitely working on it. So when you have a lot of hairy structures, hairy sort of bundle of hair, like your own hair, when it gets wet, it can close itself because of a capillary action. And we first thought that might be the case. But actually, Maddie did a really good sort of investigation on, series of investigation, which part of the papas is responsible for this closure. And actually, I mean, it does happen when they are really wet, that is hair like, bundling due to the capillary action. But when they don't even, touch each other to form the capillary action, you do start to see them close because the movement of the morphing, the papas, they actually are almost solely due to the structure at the base of the papas. It kind of acts like a hinge structure. It's a bit like a donut-shaped structure. It has a hole in the middle, and so it's a receptacle of the flower. But yeah, it has a hole inside, and then it has the tissues around it. Again, they are all dead tissue, only the cell wall remained, but they seem to have a differential expansion of one side to another, and that changes the shapes of the hinge in such a way that when it's dry, that all the bristles would positioned more horizontally, and when it's wet, they get pulled up, and the papas closes. So that's something, we would like you to stay tuned about our future reports.
Okay, thanks. A question from Taras Pasternak. What about detailed 3D images, segmentation, and 3D printing? Can we do this?
Yes. So when we wanted to make a fake papas, first thing we wanted to do was to 3D print. Well, 3D printing technology is, fast advancing, and right now, there's something that can print micrometer precision structures quite well. But even now, probably it's difficult. At least back then, a couple of years before, it wasn't the scale that we can accurately print out the real size. In real size. That's why we had to go for something like silicon wafer and then cutting it out, because another thing is where if the papas moves, like bend when they are flying, that changes the diameter of the structure too, etc., etc. And then that actually we didn't want to do, so that and the real papas doesn't bend at all when they are flying. And so we not only needed the whole, sort of a structure to be, that's sort of made in such a way, it's also as stiff and stable as a structure too. And then silicon wafer is quite strong, stiff material, and then that's what we needed to use. So yes, the size and the intricacy of the samples, the papas that we were dealing with, didn't allow usage of 3D printing, but one could scale up. If you can scale up also viscosity of media to study things. So yeah, that was definitely a possibility we were looking into. And if you want to do similar studies with plant, whole shoots and stuff, then I think that's very much possible to print out something with 3D print as well.
Great. I think one final question from Oliver Arts. So saying that evolutionarily speaking, The parachute seems quite extreme. Are there any intermediates or relatives in the plant kingdom that show a similar structure, but maybe with altered flight behavior?
And so evolutionary speaking, the parachute seems very extreme. You would think so, right? But actually, so that's the slide that I didn't get to show. I don't know if you still see, probably not, right? You're seeing the Plantae SPB announcements right now. So I'm not going to show the slide, but actually it's a quite common structural design for one type of flying seeds. So the other type is the winged seed, winged diaspore, like that of maples and pines and so forth. They have a wing and it kind of flies like a helicopter, with a leading edge vortex and so on. Yes, so that's one kind, which is more common in larger seed, heavier seed, and larger diaspore. Below the size of dandelion, or actually, more larger ones, well, like cotton and etc. too. But they are the one of the two kinds of flying diaspore dispersal unit is a hairy kind that seems to use the hair porous surface or structure to increase the drag. And the wall effect probably is playing a role in all of them. And these hairy diaspore evolved multiple times independently, and they use all sorts of different parts of the fruits and then the tissues around the fruits and then the reproductive structures to create those hairs. Yes, so they are completely independent mechanism, but they came up with the same kind of structural design and the functions that presumably they have. So I don't think it is extreme example. It's actually quite common, and it is also probably quite effective. Sorry, I didn't quite answer your question in the way you probably wanted, but I hope I did address maybe the underlying question you had.
Okay, thank you very much, Naomi, and thanks to both the speakers today for two fantastic talks, and I'll pass you back to Katie.
Yeah, thank you all for attending, and yes, thank you to our speakers, and thank you, Maddie, for moderating. That was a really interesting discussion and talk. Keeping these to 1 hour is definitely one of the struggles with this series. Be on the lookout. Within the next few days, we will be posting a recording, and we have a lot of extra questions for our panelists that we'll be emailing to them and continuing this discussion on Twitter. So thank you all for joining. Be sure to sign up for the next series in these events, and hope to see you